My name's Richard Gibbons, and I'm delighted to attend this meeting and see people I've met before, many I've corresponded with over the years. And also many thanks to Madeline Hartman and the others involved in organising this meeting, which I know has been so hard over 2 years to get off the ground. I was a young doctor when I started working on this condition. I retire next year.
No.
So it's in 1990 when I was taking a gap in my medical training to undertake some research, and I was asked if I could identify the underlying genetic cause of an unusual condition of learning difficulties. Children all had the same appearance, and they had a blood disorder called alpha thalassemia. There were only 5 of them. So we started by understanding and getting information about the clinical picture. And as ever, it's the families that tell us this information. And from 5, by 1992, we had 9 families. Families now, these were with multiple individuals affected. By 1995, we had almost 20. So at that time, we had enough families to help, like Sherlock Holmes, find where the underlying gene was. And that made a huge difference. Suddenly we could identify who carried the gene, we could provide prenatal diagnosis. And so in the last 30 years, I've been trying to work out what on earth does this protein do? And why, if there's a genetic mistake in it, does it cause disease? We're just starting. So I thought I would deal with just a few things today. It won't be a long presentation so that it can be translated. But these are the sorts of things that people have raised with me, and I thought we'd discuss. So, the way that ATR-x is diagnosed has changed enormously in the last 5 years. So, originally, we did it following the top things, which I'll go through. Remember, originally, we didn't need DNA diagnosis to make a clinical diagnosis of this condition. So, the diagnosis was suspected on the facial appearance, the pattern of inheritance, the presence of thalassemia. Now, most diagnoses are made where the diagnosis has never been suspected. It's been made by people who don't know about the condition, but by finding genetic changes. As you know, the children look alike. These were 2 of the early children that we identified. At this stage also, because the diagnosis was done on narrow definitions, we naturally saw a narrow population of children who all had very similar severity. The pattern of inheritance, of course, is very important. And the original families we saw, there were just single cases. But soon we began to see nephews and uncles related through the mother. Only boys were affected. The mothers who were carriers were unaffected. Madeline uses this image for the condition, because it's a very striking image of what the blood looks like in this condition when you stain it with a special dye. It's seen in our experience, 3/4 to 80% of, but usually that sort of number. The great thing is, if you see this, you make the diagnosis in an hour. It indicates alpha thalassemia, which is a form of anemia, but in ATR-x syndrome, it's clinically insignificant and doesn't need treatment. Now, another change which is now proving very useful is another functional test. The alpha thalassemia is a functional test of whether the ATR-x gene is working properly or not. So what is helpful to make the diagnosis is a functional test, something that tests whether the gene is working properly. So the alpha thalassemia is a functional test. In about 3/4 of affected boys, we see a chemical change to the DNA, which is a helpful functional test. And this change is called methylation. Each lane is a separate individual. The lanes come in pairs. And these are normals, normal individuals. And we've processed their DNA. And you see on the left-hand side of each pair, 1 or 2 bands, which then go down to a lower level in the next lane. Yeah. So in where there's an S, we've added an enzyme, a chemical that cuts the DNA if there's no methylation. In boys with ATR-x syndrome, the DNA doesn't cut so effectively. And about 3/4 of the boys have this pattern. And it's really, really useful for making a diagnosis. The next, I want to talk about what mutations do to the gene. So a gene is an instruction for making a protein. And in the top line, that's the instruction. The quick brown fox jumps over the lazy dog. Some mutations change the spelling and alter the meaning of the instructions. So it no longer makes sense, and the protein no longer does what it should do. So instead of fox, we have box. Some mutations shorten the gene, effectively the product. They cut it off. So we're missing the lazy dog. And in ATR-x, most of the mutations are like the middle one, like fox to box. So this is looking at the gene, which is the box along the bottom, that grey box. And each circle represents a mutation. And if it has a number in it, it means that mutation's occurred more than once. The little, in the grey box, there are some further details, like another little grey box and some grey lines. And they represent important parts of the protein that do a job. So in ATR-x syndrome, the mutations aren't spread throughout the whole gene, they're localized. Now, it's also possible to see what other changes occur in a normal population in this gene. Over 100,000 people who don't have ATR-x syndrome have had their DNA sequenced in this area. And this is where their DNA changes are. So about 1,700 of the 100,000 have got a variant DNA change in the ATR-x gene. And the changes in the normal population tend to avoid the important areas. So why am I telling you this? Most changes in ATR-x do not affect the function of the gene. Thousands of people are walking around with changes here. They don't have ATR-x syndrome. This is important because most people now get their diagnosis by finding a genetic change in the ATR-x gene. And in most cases, it's called a variant of unknown significance. In a child with learning difficulties who has a variant of unknown significance, without other evidence, they're not sure if that change is responsible for the condition. I get written to by many doctors who have a result, and this is what I tell them. How do we know whether it's pathological? Pathological means how do we know whether it causes disease? Is the clinical picture consistent with ATR-x syndrome? Have we seen it in another family that we definitely know have ATR-x syndrome? Does it fall in those two commonly affected areas? Have you looked at other males in the family to know that they haven't got the same variant? Are there HBH inclusions present? And are there DNA methylation changes consistent with the diagnosis? So even with the new technology, we can't avoid addressing the old questions. Why are so few females affected? We'll start off. All females have 2 X chromosomes. Males have 1 X chromosome. Females only use one of those X chromosomes in any one cell. And the decision as to which one to turn off occurs very early, when there's less than 100 cells in the embryo. Once the decision is made, then all the daughter cells have the same pattern. So XP, the girl has got from her dad. XM, she has got from her mum. So the female will have cells which are a mixture, some with the father's X active, some with the mother's X active. And we can see an example of this with the tortoiseshell cat. So all tortoiseshell cats are girls. One gene on the X chromosome, one flavor makes a black coat, another flavor of the gene makes an orange coat. So if the father is the orange gene version and the mother is the black version, or vice versa, because of X inactivation, the cat will have 2 different colors. Yeah. They're not a mix, it's patches. In female carriers of a mutation, and in this case, I've chosen at random for the mutation to be on the mother's X chromosome. And what happens is that when we test the mother, just one of the X chromosomes is active in every cell. And it's the paternal, the unaffected X chromosome. So the mutation isn't expressed. Mm-hmm. And the reason this happens is that if a cell has a mutation in it, it doesn't grow as fast and as easily as a cell without a mutation. So the normal cells outgrow everything. And this process protects most female carriers from the consequences of a mutation. So all of you know how common gastrointestinal problems are in ATR-x syndrome. The gut has its own nervous system, which helps propel food along its length. This doesn't function properly in ATR-x syndrome. The swallowing is discoordinated and it can lead to choking. Drooling, very, very common. Particularly, we're not sure particularly why, but it may be because they tend to keep their mouth open, their tongue out, and they don't swallow very frequently. Reflux, very common, particularly in young children, usually improves with age, and sometimes is complicated by a condition called pseudovolvulus, which I will show you. Constipation, slow transit, bloating, very common in ATR-x syndrome. Bloating probably because of the food being fermenting in the gut. Some families find taking low fermenting foods in their diet helps. Yeah. Ileus, the gut just stops. The abdomen becomes very distended, very uncomfortable. The child stops eating and will be vomiting. Sometimes the gut, particularly the large bowel, can twist on itself, and if not recognized, can lead to a bit of bowel dying. And some children have a form of a condition called Hirschsprung, where they're missing the nervous system in a patch of the bowel. So on the left, we have the esophagus that's looking like just a dilated tube. It should show contractions. The gut, the stomach should hang like this. It's twisted round on its stalk, and now it's blocked off the exit of the gut. And that means the only way for the stomach contents to leave the stomach is upwards. And that will need surgical correction. Final panel, we have what volvulus is, again, a turning of the gut. And we wonder, some of this is anatomical, but some of it is because the gut is not contracting in the normal way it should contract. So if you do a search for ATR-x on the internet, you will see more about cancer than you will about ATR-x syndrome. And that is because in the last 15 years, ATR-x mutations have been identified in some cancers. And ATR-x is thought to function as a tumor suppressor. It keeps cancer at bay. And if ATR-x is lost, that protection is lost. But almost all of these cancer-related changes have not been inherited. They've been acquired in the cancer itself. They've arisen during life. And children with ATR-x syndrome are generally not cancer-prone. But there is one exception, and that's found in the last 5 years. 5 children who were born with ATR-x syndrome have developed a form of bone cancer called osteosarcoma. 2 of those in the same family. And it presents as painful, swollen bones. But it's still rare. Treatments for ATR-x syndrome. I know this is something many of you have been very interested about. So there are treatments for symptoms, like treatments for seizures, treatments for gastroesophageal reflux or ulcers, treatments for drooling. But those are treatments that are not specific to ATR-x, and they're not curing the condition. They're reversing some of the symptoms. But more recently, there have been some developments in trying to correct the condition. A family in America have set up a foundation called Cure ATR-x. And at the moment, they're trying to raise money for treatments, genetic treatments, and they're trying to get together a panel of scientists with experience in this field. At the moment, they're raising money and trying to get interest. And they're on the ATR-x Facebook, so they'll have been in touch with you all. Gene therapy, though, is very, very difficult. Where it works well, we can take the cells out of the body, we can treat them, and then we put them back in. And that works for hematological conditions. But in ATR-x syndrome, we've got to treat every cell, particularly in difficult to reach areas in the brain. We've got to get the dose right. Too much is as bad as too little. So at the moment, that area is 10 more years away, and even then will be difficult. Drug therapy. Now this is fascinating. And this is a group in Japan who are using a drug called 5-ALA. This is a chemical that we make in our bodies every day, and there are no side effects that we know of. In Japan, it's available as a food additive. In Europe, and I think in America also, only on prescription. But it won't be available for prescription until it's been shown to specifically be effective for a condition. And currently it's not used for any medical treatments. But it was used in a mouse model for ATR-x syndrome. And when they treated the mouse, or series of mice, the mice actually improved doing certain maze tests and such like. But actually, when they treated the mouse, they expected the opposite result. They thought it would make it worse. So they don't know how it works. It was used in one case. It's not a proper trial, but the family reported that there was an improvement in language in that case. And I'll show you. This, along the bottom, is the age of the child. Just up from that in boxes is the dose of the aminolevulinic acid that was given, between 5 and 30 milligrams a day. And above that is the number of meaningful words that that child spoke. Currently, there are 23 families with cases taking 5-AAA in Japan, and 65% of the parents think it effective. But remember, that's subjective. People will see what they want to see, often in tests. Now, a very old friend of mine called Dr. Takehito Wado is running this study, and he's now got funding for a very, very small trial of 5 people. And it took a long time to get the money for that. It was very difficult to get funding for this sort of trial. But that trial of 5 individuals will start soon, where they're properly monitored and measured on objective terms for improvements. So, any questions?